This section is all about genetics and we'll be covering everything from what an exon is to the dystrophin gene itself. This can be complicated and different to experience you've had or things you've been taught. So if you have any questions, please get in contact with us. If you find these videos useful, then we have more on our YouTube channel and website for you to go through at your own pace and leisure. We encourage you to share these as much as you can. Thank you.
So, Sophia, we have discussed exons and introns in another video. We have also covered and talked about different types of mutations in Duchenne muscular dystrophy. However, what about splice site mutations in genetic sort of conditions like Duchenne muscular dystrophy? We haven't touched upon that.
Yes, basically a splice site variant is another mutation in Duchenne muscular dystrophy. It plays a significant role in the genetic disorder. These are the alterations in the specific sequence of the gene or of the spliceosome, which is basically a protein complex, and it recognizing the splicing process. This complex basically acts like a genetic scissors of our body, and it knows exactly this protein complex knows exactly where to cut. So it takes out the genetic scissors, looks at the site where it has to cut the intron out of the sequence, and it cuts in that place.
Okay, so the splicing process, if we just talk about that, so it involves removing certain parts or sequences called introns from the whole recipe, the genetic sort of transcript, we can call it, and it joins the remaining sequences together, and they're called exons, to create the final RNA or the recipe that makes protein. So we have already touched upon the fact that exons are the regions we want to keep. They are the ones who have the recipe manual that actually makes proteins, the product. And we have also touched upon the fact that introns, sometimes known as junk DNA, do not have any contribution in actually making the protein. They are important in other things in the genetic world, but not in making the protein. So before the RNA, this whole recipe manual, can go on to make a protein, the introns need to be kicked out. So there are this smart process in the body called splicing, where little scissors come in, cut out the introns, the regions we don't want, and then glue together the exons to make the final recipe manual. And it is this recipe manual that makes the protein. The splice site mutations, or what are genetic scissors, sort of sometimes are sort of genetic scissors get confused where they need to cut, can disrupt the process and lead to changes in the feeding frame. So that's another type of mutation. There's different types of mutations we discussed. One of the types is when the scissor mistakenly cuts where it shouldn't. It's supposed to cut the introns. It's supposed to cut the introns, but sometimes it cuts it in the wrong place.
Yeah, that's right. And these errors or mistakes during splicing process, they can result in improper intron removal. So as you said, the scissors get confused and they cut in the wrong place. And all this affects the whole production of the functional protein. So these genetic scissors need to accurately identify and remove the intronic sequence from the RNA, because once all the introns, non-coding elements, are removed, we get our pure exons that come together and make the protein.
But it's a challenging task for these scissors because the sequences it recognizes are short, and there are many other sequences similar to these sequences. So the correct identification of the splice site or the cutting site is crucial. And our body knows, okay, this is an intron, we need to cut it out. This is an intron, we need to cut it out. But sometimes due to mutations in the recipe manual, that alphabet that tells us to cut here can alter, and the scissors go a bit confused and cut the wrong place.
Absolutely. Splicing process is essential for proper protein making, and any disruption can lead to deficiencies in the protein level in our body. So this can result in abnormal metabolism or function, like, for example, dystrophin gene deficiency in the muscle. When the dystrophin protein is not made due to the deficiency in the gene, so our muscles not get enough dystrophin that it needs for functioning.
Yeah, and it's very interesting, we even today, we as scientists do not understand all the recipe manuals in our body. We're still trying to discover more and more about how fascinating our body is, different sort of pathways and chemical structures and things. And it's really interesting that the splicing process was first discovered in 1970s. It revolutionized our understanding of the gene expression. Like with that, it's just the understanding that the removal of introns through splice site is a crucial step in the RNA maturation. Like just the understanding that the introns need to be cut out to get the final recipe that will make the product was very, very profound. The biochemical mechanism of splicing has been extensively studied by now. There are loads of papers on it. It's characterized by the sort of cutting of introns at very specific sequences known as splice sites. And we talked about splice sites, but very specific sites. So even in the dystrophin gene, going back to dystrophin, the introns need to be kicked out. There are very specific sites where we know where to cut them out. And the most common sort of sites of these introns are at the alphabets G, U, and AG. I mean, that's just an interesting fact I wanted to give, but really I want to emphasize that these splice sites are known by the body's scissors. It goes there and cuts there, but sometimes changes in the DNA or mutations can happen that change that site. And the body can cut out, for example, an exon that we needed, or leave in an intron that we needed kicked out. So that actually changes, the reading frame, what we're trying to read, the recipe manual.
Yeah, and we have mentioned it already many times, but I can't stop emphasizing that the sequence of the genetic letters are vital, because any changes in them, they can actually inhibit splicing. For example, why this sequence is important, because we have one specific sequence that shows that this is the end of exon, and then there's another specific sequence that shows that this is the beginning of the intron, and in between we have a splice site. So this splice site tells our genetic scissors that this is the spot where they have to cut. This is the spot where it has to remove the intron in order to leave the exon in the sequence in order to make the protein.
Yeah, indeed. And it's so important to understand. And I think I was in somewhat, when I started my career in Duchenne muscular dystrophy, a bit naive about the vast sort of changes that could happen in the DNA, the mutations that could happen, until I actually met some families where we talked about splice site mutation. So their child who has Duchenne muscular dystrophy indeed carries a splice site mutation, which literally means that this scissors were cutting because of different mutations at the wrong site. So an important part of the protein was cut out that we actually needed. So I think it's really important to understand the intricacies of splicing and how mutations in the splice site, where the scissor can cut, can disrupt, and it really helps shed light on the underlying mechanism of genetic disorders like Duchenne muscular dystrophy, the complexities and the layers of complex genetics that actually occur and lead to Duchenne muscular dystrophy, or a fault in them leads to Duchenne muscular dystrophy. So it also really highlights the challenges we face, currently in terms of tackling the condition genetically, because there's so much going on. And you need to appreciate the fact that this small change is happening in trillions of cells of the body. So when you are looking at therapy, you are trying to target all the muscles of the skeletal muscle system. So that really humbles you and makes you appreciate what we are currently sort of facing.
Yes, indeed, you are right. It's a very challenging process, and that's why only through continuous research and exploration we can deepen our knowledge and to come up with better solutions, better therapies, develop the therapies. So the more we uncover about splice site mutations and their consequences, of course, the closer we get to the developing targeted therapies and intervention that for the genetic diseases that can help in the future.
Absolutely, and there's so much more to discover, and every step brings us closer to advancing our understanding for finding potential solutions, and the journey of scientific discovery never really ceases to amaze me. And I think we are further than we've ever been in terms of finding an effective therapies to tackle Duchenne muscular dystrophy, which is quite reassuring, and something that gives us a ray of hope.
Yes, absolutely, Marine.
Here at Action Duchenne, we are committed to inform, but also support you through your Duchenne journey. If you'd like to talk about anything raised in this video, please feel free to contact us on 07535 498 506, or alternatively, email us at info@actionduchenne.org. Thank you.